So, welcome students, welcome to a new lecture series of the ongoing series that is Introduction to Microbial Diversity. So, in this lecture, we will be starting off with the early classification attempts. My idea behind this making this video is that before going directly into the five kingdom classification system or the Carl-Lewis system, I want to give you a brief knowledge or a brief idea about the early classification attempts so that you can understand that what was the need or what was the requirement for um, behind the five kingdom classification system, or if you say behind the three domain system of Carlos. So, what was the requirement actually? So, before understanding the requirement, you have to understand that many people have been trying to classify the organisms, classify the microbial diversity before this five kingdom came into play or this three domain came into play. So, this lecture will give you a detailed knowledge about the early classification attempts. So, let's start off with the first person who tried to classify the organisms. The first attempt was done by Aristotle. So, in the period of 384 to 322 BC, the first attempt at classification was made by Aristotle. He basically did was to divide the living organisms into plants and animals. Okay, he basically divided on the basis of plants and animals, and the criteria on which he divided was that it did not move, got nutrition from soil. So the organisms those did not move, he basically kept them into the plants, and the organisms that could move and just ate the prepared food, he kept them into the category of animals. So this was very simple, and there was no place for microorganisms. So obviously this system got omitted out because there was no particular class where organisms could microorganisms could be placed, because at this point of time this microorganism concept was uh, completely unknown. So the limitation was obviously it was too broad, many organisms didn't fit well, so automatically the system got omitted out. The second attempt was by Carolus Linnaeus. This person also identified or introduced the binomial nomenclature system. So he was known as the father of taxonomy, and he introduced the binomial nomenclature system, that is the genus and species concept. And coming into the classification of his contributions, he proposed a two kingdom system. What he did was that in two kingdom system he similarly kept plants and animals and called it as plantae and animalia. In plantae, what he did was that he kept all the kinds of plants, and he also included some algae, some fungi, and some bacteria. At this point of time, these were coming into action, so he kept them into the group of plantae. Then what he did was that in the next one, that is the animalia, he kept all animals including protozoa. So he kept protozoa into this animalia group, animalia kingdom. Okay. But the limitation which he faced was that many unrelated organisms were placed together. For example, the first one is the bacteria and plants. We cannot place bacteria and plants into the same kingdom, because of course if we talk about bacteria is bacteria is they can sometimes they can move, or they can be unicellular. You can say they can be unicellular. This is a nice thing. They are unicellular, these are multicellular. So here obviously the point of keeping them in the same thing omits out. So the mode of nutrition, cell wall structure, these are also different. So completely this concept was wrong. We can't keep bacteria and plants together. So again, this system was omitted out from the classification system. Now, let's move on to the third one, that is the Ernst-Haeckel system, which was introduced in the year of 1866. What he did was that he kept Plantae and Animalia, but he introduced a third kingdom, that is Protista. Now, these Protista, what are these Protista? These Protistas are nothing but unicellular organisms like protozoa, algae, and bacteria. Okay, as I said that Plantae is multicellular. They are always multicellular. So if they are multicellular, there is no place for unicellular organisms. So where will these unicellular organisms go? So these unicellular organisms will basically go into the group of Protista. Now again a limitation came into play. What was the limitation? Still grouped bacteria with eukaryotes. Now, who are the eukaryotes? These protozoa are the eukaryotes. Now, you can't place prokaryotes, those are bacteria, and eukaryotes into the same group. No, you can't do that. It's completely um, illogical to keep these two in the same kingdom. So, again, this system got omitted out. Now, the second, the fourth attempt came into play, that is the Copeland system. This was introduced in the year of 1938. So, in the year of 1938, Copeland, what he did was that he proposed a four kingdom system. Now, let's see what he did was. We are aware of these three systems, kingdoms: Animalia, Plantae, and Protista. But what we see is that Monera, this is a system which he introduced. He added Monera. What was the speciality of adding Monera into the kingdom? This Monera consists of all prokaryotes. That can be bacteria, that can be cyanobacteria. Cyanobacteria are blue-green algae. So it contained bacteria as well as algae. So this was completely made for all the unicellular organisms. Okay, are you getting my point? What I'm trying to say? He placed all the unicellular organisms into the Monera kingdom, and Protista contained of protozoa, which are eukaryotes, Plantae are the plants, and Animalia are the animals. So this Copeland system was somehow or somewhat accepted, partially accepted. I can say it as partially accepted, until and unless Whittaker's five kingdom classification came into play. Now, what impact did this do? This person, R.H. Whittaker, in the year of 1969, proposed a famous five kingdom system. Now, let's see what extra did he added. Okay, now we can see he added fungi into his kingdom system. So this is where it changed from 4 to 5. Okay, so he added fungi, because see, fungi has a different kind of cell wall structure. The cell wall of fungi, if I give you a detailed or brief short note, the cell wall of fungi is composed of chitin. This is a component. This is particularly present in the cell wall of fungi. But the cell wall of plants is composed of cellulose. So this is the reason why fungi has to be placed into a separate kingdom. This was the reason behind placing fungi into a separate kingdom and making fungi as a separate kingdom. So this was the best thing Whittaker did. Whittaker made these 5 kingdoms and he exactly placed fungi into a separate kingdom because of its cell wall structure, because of its typical cell wall structure. So what it is saying is that based on the cell type, either prokaryotes or eukaryotes, based on the cell number, either unicellular or multicellular, and based on the nutrition mode, that is autotroph or heterotroph. These were the factors which he kept in mind until huge improvement came until Use's system. So this was accepted worldwide. This 5 kingdom classification system was worldwide accepted until Carl Use's system came. Okay. So now we will understand about another thing, a term called binomial nomenclature. So already I have given you a lecture, already I have told you about what is binomial nomenclature. So as I told you about Carl Linnaeus, who has introduced this binomial nomenclature system. So binomial nomenclature is a formal system for naming species. Each species is given a two-part scientific name which consists of genus and specific epithet. For example, the name of Homo sapiens. This system was popularized by the Swedish botanist Carolus Linnaeus in the 18th century and now is a fundamental part of taxonomy. So here in this slide, there is nothing much to introduce to you. You already know what is binomial nomenclature. I have just included this slide because I have told about Carolus Linnaeus, and he has introduced the system. So I have just included this slide. Over here also, you must be knowing that it is a two-part name where we use genus and a specific epithet or a species name. It is Latin in origin, and always a capitalization is used in the genus name. When we are writing the genus name, the first letter is capitalized, while the specific epithet or the species name is never capitalized. Okay, so these are the key features and rules. And coming into the italics or underlining, when we are typing it, we can type it in the italics form, that is Homo sapiens, but when we are writing it manually, then we have to underline it separately. Okay, example Homo sapiens, like this. Uniqueness, what it is telling, each species has a unique binomial name. Of course, all species have a particular binomial name, and this is the only reason why scientists use this binomial nomenclature system to identify the species. So apart from using the common name, scientist always uses this binomial name to identify a creature or an organism. This prevents the same name from being used for two different species. Okay. Now abbreviation, what it is telling? It is telling that once a scientific name has been mentioned in full, the genus name can be abbreviated to its first letter, such as C. lupus. Okay. Like for instead of writing Canis lupus, you can write C. lupus also. Another thing we have to keep in mind, it can come as your short questions, that the international codes of nomenclature are International Code for Zoological Nomenclature, that is ICZN. So ICZN is International Code of Zoological Nomenclature. ICBN is International Code of Botanical Nomenclature. These are the nomenclature codes, international codes, for, and they govern the rules for naming animals and plants respectively. And for in the case of prokaryotes, in the case of bacteria, earlier this was called as bacteriological code. This is called as International Code of Nomenclature of Prokaryotes, that is ICNP. This basically governs the naming of bacteria and archaea. And last but not the least is the ICTV, which is the International Committee on Taxonomy of Viruses. So viruses, naming of viruses is done by this nomenclature, this system, ICTV. Okay. So what are the advantages of binomial nomenclature? If I come into the advantages of binomial nomenclature, now obviously the first one will be the universality. The second one will be the precision. The third one will be the simplicity and standardization. So, universality, the same scientific name is used all over the world and it avoids confusion caused by local or common names. Example, Mangifera indica. If you tell mango, it is recognized globally, and Mangifera indica is the scientific name given to all mangoes. Now, precision is that each species has a one unique name. Like if the species differs, the species name will also differ. It helps in avoiding mistakes in identifying the organisms. Simplicity and standardization, it always uses two words, genus and species. It doesn't carries other words, so it avoids any kind of misconceptions or any kinds of um, what to say, any kind of complexities. And it is very easy to remember and follow compared to long descriptive names. Okay, compared to long descriptive names, it is easy to follow up. It also shows a relationship, that is, the genus name shows a close relationship between species. For example, Homo sapiens and Homo erecticus belong to the same genus Homo. So we can understand that this species and this species is almost related to each other because the genus is same. Then it gives stability. Names are given by rules of international codes such as ICBN, ICZN, ICPN, whatever. Once accepted, names are stable and not frequently changed. You can't change the name frequently whenever you wish to. And it facilitates research and communication. Like it helps scientists and students and also researchers to communicate clearly via these established names. It avoids mix up. For example, if I call panther, it can mean as leopard, it can mean as cougar, jaguar in English. But panther means Panthera pardis. It is only the leopard. So if I talk about leopard, it is only Panthera pardis. It will omit all these names. But if we call panther, then this panther can mean it can be leopard, it can be cougar, it can be jaguar. But if I tell Panthera pardus, then it will mean only leopard. Are you getting my point? What I'm trying to say? So, here, just let me check once. So, if I start off with the Whittaker system in this particular lecture series, I think so that will be a bit difficult for you to grasp. So let's keep it clear, cut, and simple. Let's keep this lecture up to the binomial nomenclature. And starting off with the next lecture, I will be dealing with the Whittaker's five kingdom classification, and then eventually I will move in into the Carl user system. So, till now, I have taught you till the binomial nomenclature and the early concepts of um, early attempts of classification. So again, I would say that if you like my videos, do like, comment, and don't forget to subscribe to the channel to get regular updates on whatever I'm posting into the channel. As this is a completely new channel, this channel would be much more efficient with the help of you people. If you guys like my contents, then do like, and do don't forget to comment on which video I should be making the next. Then I will try to make the videos on that topic. And this is the official website where you can go and check out the batches that are being started. And this is the number where you can call or WhatsApp for admissions, because online batches are being conducted. So you can obviously go and take and enroll at your name into this academy. So till then, take care, and see you in the next lecture. Okay.